Blood types are a very important concept within the health field. There are two major types of blood types that we're going to take a look at and compare now: the ABO blood type system and the Rh factor blood type system. Before we get into those specifically, let's talk about the surface of cells, in this case red blood cells specifically. When we're first learning about cells and we're seeing the shapes of spheres with nuclei and learning all the parts, we tend to just picture round cells. And we also tend to picture them as a very smooth surface. And so I have an analogous picture here. We often think, kind of like the surface of the Earth here is, here's the surface of what a cell might look like in large scale, just very flat plasma membrane. In reality, there are a lot of chemicals sticking off the surface of our cells into the extracellular space. And so if we were to actually draw this analogy to what a real cell looks like relative to the ground, instead of being smooth and flat like this, it would probably look a little more like this, with all the trees and plants that you see representing different chemicals that are actually sticking off the surface of the cell. We looked at this a little bit earlier in the course when we were looking at the plasma membrane of the cells. We looked at images like this, where we had the phospholipid bilayer, and then we had proteins embedded within the phospholipid bilayer, as well as carbohydrate chains sticking off the side of those proteins and out into the extracellular space. Even this drawing that we have from our textbook is an oversimplification of what the cell would actually look like. But for now, we just want to focus on the ABO system and the Rh factor system. The ABO system is actually carbohydrate chains that are sticking off of the plasma membrane as shown in the image here. The Rh factor is actually a type of protein that is found on the surface of the red blood cells, just like shown here. So let's look at these systems. For the ABO system, we have type A blood. Type A blood has a certain form of the carbohydrate, in this case represented by a square, that ultimately sticks off the red blood cell. In type B blood, those with type B have a different type of chemical sticking off the surface. In this case, we've represented it by a circle, and as you'll note here, we're talking about A antigen and B antigen. The term antigen is just referring to a chemical found on the surface of the cell. A person with type AB blood actually has both A and B antigens found on the surface of their cells. A person with type O blood has no antigens of this type found on the surface of the cell. Going back to the analogy I was making of the Earth earlier, don't think of type O as having no chemicals on the surface. It just doesn't have this specific type of carbohydrate on the surface. It would have plenty of other chemicals that we're not representing in this image here. So what we need to think about is, for someone who has type A blood, their body is used to having type A antigen on the surface. They wouldn't want to have an immune reaction to their own body, and so they will not make antibodies, which ultimately fight off infection and antigens that are not recognized as us. And they do that through antibodies that will recognize other chemicals. In this case, what we're showing for type A here are the antibodies in that person's immune system that will be produced if type B blood is donated to a person who has type A blood. That person with type A will recognize that this chemical, B antigen, ought not to be in my body. It's something foreign, and so I will make an immune reaction producing antibodies against it. A person with type B blood will recognize the B antigen as themselves, but then if we put type A blood into their body, they would produce anti-A antibodies, which would then go and attack the type A blood. A person with type AB blood will actually have no antibodies to either type A or B, because if they did, they would actually attack their own blood, because their cells have A antigen and B antigen on the surface. Type O has no antigen on the surface from the ABO system, and so they produce both anti-A and anti-B antibodies. And so if you were to put any of these other blood types into a type O person, they would actually form antibodies and attack that blood type. The Rh factor again, is a type of protein found on the surface of red blood cells. And when analyzing the Rh factor in blood types, it's a little bit simpler than the ABO system. Someone who has positive blood for the Rh antigen has the chemical on the surface of the cell. Someone who is Rh negative does not have that chemical on the surface of the cell. Again, they have other chemicals, but they do not have the Rh factor. I've shown the antibodies here, indicating that somebody who's Rh positive will not make antibodies against the Rh factor, otherwise they would attack their own cells. Someone who is Rh negative does not have that chemical in their body, and so they will produce antibodies against the Rh factor if you were to give them positive blood. These are both important concepts so that we can explain the relationship between blood type and blood transfusions. Not all people can accept blood from every other blood type. What we need to be able to do is explain the relationship and be able to analyze whether a blood transfusion would ultimately be successful, or if the recipient's body would produce antibodies to then attack the blood of the donor. What we could do is look at questions like, can an A negative person give blood to somebody who is AB positive? What we ultimately need to analyze is, will the person who is AB positive produce antibodies and attack the A negative blood? If that's the case, then the blood transfusion is useless, because if the AB positive person is going to attack the A negative blood, then when you give them the transfusion in the hospital, the person with the AB positive blood is no better off because their immune system will start to react to kill off the A negative blood as if it is a bacteria or a virus, some sort of pathogen that has gotten into their body. How do we go about analyzing whether this transfusion would be successful? One of the keys is to analyze the ABO system and the Rh factor system separate from each other. What we really need to do is ask the question, can A blood be donated to an AB recipient? Can Rh factor negative blood be given to positive blood? If we're receiving the blood as an AB blood type, we have A antigen and B antigen on our surface. And so we will not make any antibodies against A to attack it or antibodies against B to attack it because our immune system will recognize those chemicals as part of our body. If we're given blood from somebody who is type A, they have A antigen on the surface. No antibodies will be made by the AB person against the A antigen, otherwise they would be attacking the A antigen on the surface of their own cells. In the end, an A blood type person can donate to an AB recipient. Can we give negative blood then to a positive person? The recipient, being Rh positive, has the Rh factor on their surface of their cells. The donor has no antigen on the surface. So the Rh factor recipient has nothing to actually attack in terms of the Rh factor on the negative donor. And so, in the end, this is an acceptable transfusion as well. Because the A can be donated to AB and the negative blood can be given to a positive person, overall we can say this transfusion is acceptable. An A negative person can donate blood to an AB positive person. This makes it much simpler than trying to memorize, can an A negative give to an AB positive, or can an AB negative give to an O positive person, and trying to memorize all the potential configurations that could happen. Break it down and it becomes a lot easier.